So I'm very excited to tell you a little bit more about my postdoctoral project working on transcriptional regulation, often by transposable elements, during mammalian preimplantation development. And I'm super interested in transposable elements, as these are these regulatory sequences that are scattered throughout the mammalian genome. And actually, over half of our genome consists of remnants of transposable elements, as you can see here in this snapshot of the mouse genome. And these mobile elements were originally transposing and multiplying in the genome, but they became mostly dormant over time in mammals. So this means that at time of insertion, they contained the coding sequences or the sequences that allowed them to transpose, either retrotranspose in the case of the retrotransposons, the LTRs, lines, and signs, or using the transposase mechanism of DNA transposons. However, over time, due to multiple generations and silencing of the host genome, a lot of these transposons lost the sequences that are necessary in order to transpose and multiply. However, they still contain transcription start sites as well as transcription factor binding motifs that allow them to become transcriptionally active. And this is interesting because these transposable elements are typically silenced in somatic cells, but they maintain the ability to become expressed. And this is particularly studied in the context of early embryo development, which is what I'm going to talk about today. But it's also very interesting in the context of stem cells and cell fate, as well as the untimely activation of transposable elements in cancer. And so there's this hypothesis that these once selfish elements became co-opted by the host genome for regulatory purposes. And so we know quite a bit about both genic and transposable element expression in the early mouse embryo. So we know that in the mouse preimplantation embryo, the genome becomes activated, so highly transcriptionally active, and takes over the maternally inherited transcripts in the early to late two-cell stage. So this is when really the zygotic genome transcriptional program kicks in. And this is preceded by a minor wave of transcription as well. And this coincides with large epigenetic remodeling, DNA methylation status changes, histomodifications needs to be reestablished, as well as the rise of genome architectural features post-genome activation. And this coincides also with the transcriptional activation of several transposable element families that we know quite well in the case of the mouse. And although at first you might think that this is simply a consequence of this large transcriptional activation of the zygotic genome, as well as these epigenetic changes, however, it has been found that these transposable element activations are really hallmarks of the preimplantation embryo. And when disrupted, it affects proper progression of embryonic development. And so, of course, transposable elements are not unique to the mouse. They can be found all throughout life. And so we were wondering, are patterns of transposable element expression shared across species? And then what can we learn about these conserved as well as species-specific genome regulation rules by transposable elements? And so in order to do this, we focused on the preimplantation development of the mouse, pig, cow, rabbit, and rhesus embryo. But before I'm going to dive into the data, I'm going to highlight just one difficulty when working on transposable elements. First of all, they can be very tricky because they are repetitive by nature. But the second reason why they are difficult to work with is because they can be very close or even within genes. And so this means it can be very difficult to figure out whether a transcript is a result of a read-through transcript where the genic transcription start site is driving the expression of a given transcript and the TE sequence is simply read through as the polymerase amplifies it, or whether it's a TE-driven transcript where it's really the TE sequence that is initiating the transcription. And so at the onset of this project, Diego, my co-first author on the paper, decided to develop a new technique that is able to differentiate TE-driven transcripts from genic read-through events, especially when working with low input samples, which is what is the case in our study since we work with the embryo. And of course, the main difference is whether the 5' lands inside a genic sequence or whether the 5' is really truly mapping to the TE sequence itself. And so he based the technique, which we call SmartSeq +5. He based this on the SmartSeq2 RNA-seq approach, which uses transposase to capture and prepare the library of gene coverage. And before this transposase, you do a reverse transcription, and Diego added a step where we now add a little barcode in front of the 5' Nextera adapter, which allows us to separate now, after the second step, the internal fragments, which will have the canonical Nextera adapter, from the 5' fragments that will have this little extra added barcodes. And then after sequencing, we can computationally separate the internal fragments, here in this example in the actin gene, that nicely cover the gene, from the 5' fragments that indeed captures exactly the transcription start site. And so for today, I'm just going to focus on the results based on the 5' fragments, but of course our data contains both. And we identify TE-driven transcripts and the transcription start site profile based on where the 5' of the reads fall. And so we did this, as I mentioned, in the pig, cow, rabbit, mouse, and rhesus pre-implantation embryo. And before I truly go into the data, I just want to highlight that the timing of genome activation, so this big burst of transcription that we see in the mouse 2-cell stage, occurs at slightly different timing in the different mammalian embryos. So for example, in the rhesus and also in us humans, this happens in the 8-cell stage. So throughout the presentation, I will highlight in yellow the timing of embryonic genome activation. Okay, so on to our actual data. So here I'm plotting, first I was interested what happens on a general subclass level to the expression of retrotransposons, so the lines, LTRs, and signs. And when you compare the mouse to the other mammalian species, you can see there are some conserved patterns of expression. For example, you can see that LTRs typically show an increase of expression right at timing of genome activation across all of our species. However, when you look at the other retrotransposons, for example, the lines, you see that a much more species-specific expression profile. And I just want to highlight that this is not caused simply by genomic frequency of these elements. For example, if you again look at the lines, this is nearly not expressed or visible compared to the overall expression level of the LTRs and signs in the mouse. However, when you compare in the rhesus, the lines become much stronger expressed, but they have similar genomic frequency in the genomes of the mouse and the rhesus, suggesting that it's not simply explained by the frequency of these transposable element families. And the other thing that I was highly interested in is understanding more about the evolutionary age of these transposable elements. So here I'm plotting based on whether they're shared across, for example, the orders in purple. So this in mouse, this would be a mus shared transposable elements. Whereas, for example, in red, these are eutherian shared transposable elements. And this means that if it's eutherian shared, these transposable elements were originally inserted and multiplied in the genome of a common ancestor, and meaning that they're over 99 million years old. Whereas the mouse-specific transposable elements are only 14 million years old. And this is interesting because if you think about transposable elements as selfish elements, you might think that these very young transposable elements become higher expressed than the older ones, as the older ones have had more generations to be silenced. So is this indeed the case? So here I'm going to plot first just the cow over evolutionary time. So on the top, the very old transposable elements, and in the bottom, the younger order shared transposable elements. And the size of the dot will represent the frequency of expression. And as you can see here, in the case of the cow, indeed, there are some of the younger transposable elements that still that are expressed, but we also see very old transposable elements that remain expression in the cow pre-implantation embryo. And again, this is not simply something that can be explained by frequency of these elements in the genome. As you can see, for example, the cow has a lot of order shared younger signs in the genome, as well as some very old mammalian shared signs. But when you look at their expression level, they become expressed to a similar extent in the cow embryo, suggesting again that it's not simply explained by the frequency of insertion. And this is something that we see across our panel of species. We see many ancient transposable elements becoming expressed. even though they've had many, many million years to become silenced and become repressed or even completely lost in the genomes. And only mouse that have also a high frequency of these younger transposable elements in their genome show a very strong expression of the younger transposable elements. And so the next thing I was interested in is not just to look at this very broad level, but specifically which transposable elements become expressed at timing of genome activation. Because I already showed you this plot that the LTRs in the mouse show a striking increase of expression right at timing of genome activation. However, if you plot all the individual transposable element families as a heat map, so here every row is an individual family of transposable elements, you can see that there are several different patterns of expression. You can see indeed a cluster of transposable element families that become expressed right at timing of genome activation, but you also see a subset that is maternally inherited and unique to the oocytes, and another subset of families that becomes expressed prior to genome activation. So for the sake of this example, we're going to look into this cluster 6, which contains the families that are specifically expressed at timing of genome activation. And when we then again represent the individual families as rows, now representing their RPM value, you can see that the vast majority of the signal originates from this MT2MM family, which is a very well-known hallmark of development in the mouse embryo, and indeed specifically becomes expressed at timing of genome activation. And so I did this selection of genome activation transposable element families across LTRs, lines, and signs in all 5 species. And this led me to a list of 10 to 12 genome activation TEs per species. And so next, the thing that I was interested in is to understand if any of these transposable element families are actually shared across the different species. And you can see that there are quite a few that are shared with one prime candidate, which is MLT1A0, which I selected in the pig, cow, rabbit, and rhesus. And indeed, you can see that they have a similar temporal dynamics in the preimplantation embryos of these different species. They typically see an increase of expression right at timing of genome activation. And in some cases, you also see a maternal load of this MLT1A0 transcript. Now, you might wonder, MLT1A0 is present in these 4 species. Does it also get expressed in the mouse? And indeed, we do see expression of MLT1A0 in the mouse, although it is about a 10-fold lower in RPM compared to the other species. And this is quite interesting because MLT1A0 is a Eutarian LTR, meaning that it was indeed inserted about 100 million years ago in a common ancestor. And as the genomes of the different species diversified, also MLT1A0 co-evolved with its host. And suggesting that we find MLT1A0 expressed in the different species, even though there were so many generations and so many million years to completely suppress the expression of MLT1A0. This suggested to us that maybe MLT1A0 is co-opted for some kind of benefit to the host. And so I set out to understand better how MLT1A0 could be co-opted by the host genome and how this is maybe shared across species as well as maybe perhaps different in, for example, the mouse. And so there are many different ways of co-option by transposable elements. There are many examples known. However, we've decided to focus on co-option of transposable elements as alternative promoters, as this is something that we can easily find in our SmartSeq plus 5 data. And so an alternative transcription start site would result is in our SmartSeq plus 5 data as a chimeric TE gene read, where the 5' of the read would fall inside a transposable element sequence, and then the other end of the read would fall inside a genic sequence. And this is, of course, different from a canonical genic read, which would both have the transcription start site as well as the end of the read within the genic sequence. And this is something that we actually find across our different species in very large numbers. So we find thousands of examples of these chimeric transposable elements, so the use of alternative promoters of these TE sequences. And we also find all subclasses represented. So we find alternative promoters, LTRs, lines, signs, and even DNA transposons being used as alternative transcription start sites. So we wanted to know better, and specifically looking at our example of MLT1A0, how these TE-driven chimeric transcripts are expressed. And so looking at the example of C1d, which is a nuclear co-receptor, we find that indeed in our data we can find the canonical transcription start site, but we also find a known alternative transcription start site. And most interesting to us, of course, we find that an MLT1A0 insertion is used as an alternative transcription start site. Now you might wonder, how frequently is this transcription start site used compared to the canonical? And indeed, we find that the canonical transcription start site is used across all the different stages in the pig embryo. However, the MLT1A0 transcription start site has a specific increase of usage right at timing of genome activation, suggesting that MLT1A0 allows for a more temporal resolution right at timing of genome activation. And this is not specific to the pig. We also find example of MLT1A0 insertion being used as a C1d alternative transcription start site in the cow, where again, it is specifically used at the timing of genome activation. And interestingly, when we look at C1d in the mouse, we don't find MLT1A0, but we found another transposable element, MT2CMM, being used as an alternative transcription start site, and again, reflecting this increase of expression and increase of use of this transcription start site right at timing of genome activation in the mouse. And this makes total sense when you look at these transposable element families as a whole. MT2CMM has about 4,000 insertions, but all of those together show the same temporal resolution right at the timing of genome activation, whereas I already showed you MLT1A0 in the pig and the cow also has this increase of expression right at timing of genome activation. And so taking MLT1A0 out of this genomic context and more in an isolated case, we were wondering, can MLT1A0 really properly drive the expression of a gene? And to test this, we put MLT1A0 in front of a Ruby reporter, or as our negative control, we didn't use any promoter at all. And we injected it in the zygote, in both the mouse and the cow zygote, and we waited till genome activation to read out the Ruby signal. And indeed, in the cow, just like as our C1d example, MLT1A0 can drive expression of the Ruby when in the 8-cell stage. However, when we put MLT1A0 in front of Ruby in the mouse embryo, just like in the case of C1d, there's no MLT1A0, we don't see any Ruby signal in the mouse 2-cell embryo. But interestingly, when we use MT2MM, a MUS-specific younger transposable element in the mouse, we see that instead MT2MM can drive Ruby expression in the mouse 2-cell embryo. And so just to summarize what I just told you, and I've only, in the interest of time, showed you some highlights of our study, but I showed you that we developed SmartSeq +5 to accurately capture transposable element expression and TSS positioning in single embryos. I showed you that both young and surprisingly also ancient transposable elements become expressed throughout early mammalian development. And then we focused on the example of MLT1A0, which can drive gene transcription in the cow embryo, but not in the mouse. And I highly encourage you to have a look at our data sets. We mostly focused on transposable elements, but of course, we also have genic information in our data. And we made our data available as a Shiny app, which allows you to plot your favorite gene or transposable element in your species of interest quite easily. And of course, we also have our data available on Geo. And with that, I would like to thank everyone in the Torrespedia lab, especially Maria Elena and Diego, my co-first author, as well as all the collaborators that allowed us to use the different species and the different embryos, and the computational support from Henrik and Jonathan and their labs. And with that, I would like to thank all of you for your attention. I'm happy to take any questions.
Thank you very much, Marlies. Very interesting. Very cool work. Yeah, while we wait for some questions to roll in, I can maybe start. So I was really excited or interested to see that you also have these kind of ancient transposons as well as the young transposons being activated, which I guess you were also surprised by. But is there any kind of species specificity to whether they're activating a lot of young or old? Like, do you see any, any difference in that regard?
So mouse is always a bit of an outlier.
Mm-hmm.
So I'm sorry for all the people using mouse as a research model, but we find in our data set that mouse is an outlier, at least in the transposable elements regards. Mouse has a lot of younger MUS-specific transposable elements, and they also show the most expression. And also when we look, for example, MLT1A0, we see that a lot more of the MLT1A0 insertions that do exist in the mouse genome are lost their integrity, lost their transcription, and it's been taken over by the younger mus-specific transposable elements. Now, for the other species, we do find a lot of very old transposable elements. Sometimes we find species-specific co-option of these transposable elements that are much older. So they are shared across species, but not all species use the transposable elements the same way. And this is something that is super interesting to me. We haven't had much time yet to explore all the fine details yet, but I mean, the co-evolution of the host and the transposable elements is definitely something that I'm planning to venture out in the future as well.
So Uri Frank asks, are transposons being expressed in adult stem cell differentiation?
So they are indeed some stem cells that do still show expression of transposable elements. They're not typically these kind of transposable elements. So these families are typically specific to the embryo transcriptional program. But there are, for example, indeed some human blood stem cells that do still have specific transposable element families that do become expressed and are unique to these kind of stem cell systems. I mean, I told you that half of the genome is transposable element sequences, but not half of the genome becomes expressed in the embryo. But the hypothesis in the field is that these transposable elements kind of act as regulatory platforms that allow for transcriptional program switching. And they are also important in different stem cell settings.
Naveen Ramakrishna asks, curious about the translatability of the chimeras. In the Ruby example, but also the C1D chimeric examples, does the TE gene transcript sometimes impact the gene coding sequence, like alternative ORFs or starts? Sorry, it's a long question.
Yeah.
Can you see it?
Yeah, so we didn't really explore sufficiently enough to give you a clear answer on that. So we do often see that the first exon gets skipped, but we didn't do any proteomics to see if actually the protein changes. Does the protein actually get the transcript actually gets translated? So we don't know much about that in this case. But if you're interested in that, like 2 years ago, there was a study by Andrew Motzaleski who looked at another TE chimera, the CDK1 chimera that is very important in mouse embryo development that also uses a TE chimera as an alternative transcription start site. And they did much more exploration of how this alternative transcription start site led to a different protein coding sequence. And so this is quite interesting, but in the scope of our project, we didn't have time to really dive into the details of that.
Noel Lowndes has asked a question that's somewhat similar, I guess. Any evidence for transposon-induced transcription resulting in a variant transcript or protein being produced, or is it just an overexpression mechanism to boost the normal protein product during development?
Yeah, I mean, yeah, this is something we don't know in our system. So for now, we just see it as a boost of transcription, and it could just act as an enhancing to general transcription. We don't know what it actually does down the road. But yeah, this would be super interesting to follow up on.
And then Alex has asked, can you tell us more about the evolution of TE dormancy, i.e. the loss of genomic movement?
So I don't, this is a very broad question, so I have a lot of things to say, but I will keep it a little bit brief. But this really depends also on the TE family, how they evolved with their host genome over time. So you can find examples of many different TE dormancy and how they got silenced. In general, you typically see that TEs lose the sequences that allows them to transpose. So this is particularly interesting and relevant to the LTRs that typically lose the internal sequences and maintain only the LTR, the promoters of the sequence, and the lines that also lose the internal protein coding sequences. In the case of genome evolution, something that I was very interested in and that we looked at already a little bit is, for example, where is MLT1A0 located in the different species? So do they still maintain kind of orthogonal regions where we can find them? And we do find that there's, when you do liftover between the different genomes, that the positioning of these MLT1A0 sequences, especially the ones that become expressed, are in similar genomic neighborhoods between the different species. But yeah, this is very different answer for different TE families. So yeah.
Okay, I think that's all the questions we have for now. Okay, well, let me thank again Marlies and also thank Anzi for two really, really great seminars.